Growth in plants is of two types, primary growth and secondary growth. Primary growth is universal. Primary growth takes place in almost all of the plants. The growth in linear manner, the growth in vertical manner is called as primary growth, which is the universal in almost all the plants. Growth in diameter, also called as width. Growth in diameter, also called as width, is called as secondary growth. So growth is of two types, primary and secondary. Linear growth is primary, growth in diameter or width is secondary. Primary growth is universal, but secondary growth is not universal. Secondary growth occurs in some plants only. Secondary growth occurs in dicot stems, secondary growth occurs in dicot roots, and also secondary growth occurs in gymnospermic stem and gymnospermic roots. Altogether, secondary growth is present in dicots and gymnosperms. All angiosperms do not have the secondary growth. All angiosperms do not have the secondary growth. Dicots and monocots, together called as angiosperms, only dicots are having the secondary growth. Monocots do not have the secondary growth. Gymnosperms, which are the primitive to angiosperms, they are also having the secondary growth. Growth in diameter or width, that is called as secondary growth. That secondary growth takes place in dicot stem, dicot roots, gymnospermic stem and gymnospermic roots. In monocots, secondary growth is absent. In monocots, secondary growth is absent. Now I am going to explain the secondary growth in dicot stem. So if you see the primary structure in dicot stem, if you see the primary structure in dicot stem, U-stele is present. U-stele means 15 to 20 vascular bundles present in a ring-like fashion. 15 to 20 vascular bundles are present in a ring-like fashion. This type of stele is called as U-stele. This type of stele is called as U-stele. So many vascular bundles, round about 15 to 20, are present in the form of a ring, that is called as U-stele. Let us see, this one is the xylem and this one is the phloem. Xylem, phloem, xylem, phloem, xylem, phloem, and xylem and phloem. In each vascular bundle, between xylem and phloem, cambium is present. This cambium is called as intrafascicular cambium. This cambium is called as intrafascicular cambium. The cambium which is already present inside the vascular bundle is called as intrafascicular cambium. Intrafascicular cambium position is lateral. Lateral in position, whereas origin is primary in origin. Position is lateral, origin is primary, function is secondary growth. The cambium which is already present inside the vascular bundle is called as intrafascicular cambium. Intrafascicular cambium is lateral in position, primary in origin, function is secondary growth. Now, in between vascular bundles, primary medullary rays are present. In between vascular bundles, primary medullary rays are present. The parenchyma present in the primary medullary rays undergoes dedifferentiation and produce another cambium primary medullary rays undergoes dedifferentiation and produce new cambium. That new cambium is called as interfascicular cambium. The cambium which is newly formed from primary medullary rays, the cambium which is newly formed from primary medullary rays by dedifferentiation, that cambium is called as interfascicular cambium. Interfascicular cambium is also lateral in position, that is lateral in position, but origin is secondary. Because it originated from primary medullary rays by a process called as dedifferentiation. That's why it is secondary in origin. Position is lateral, origin is secondary, function is as usual secondary growth. Altogether, now we are having two cambiums, intrafascicular cambium which is already present inside the vascular bundle and interfascicular cambium present in between vascular bundles. Interfascicular cambium that is present in between vascular bundles. Intrafascicular cambium, interfascicular cambium. These two cambiums combined and formed a ring. That ring is called as vascular cambium. Vascular cambium formed by the fusion of intrafascicular cambium and interfascicular cambium. Both are combined and formed vascular cambium which is responsible for intrastellar secondary growth. Vascular cambium is responsible for intrastellar secondary growth. Secondary growth is of two types. First inside the stele, second outside the stele. Inside the stele, the secondary growth is called as intrastellar secondary growth. Outside the stele, the secondary growth is called as extrastellar secondary growth. Vascular cambium which is formed now by the fusion of intrafascicular cambium and interfascicular cambium, that ring is called as vascular cambium. Vascular cambium promotes intrastellar secondary growth. Vascular cambium, that is the ring, this vascular cambium divides periclinally and mitotically. Cells of vascular cambium divides periclinally and mitotically and produce more cells towards inner side, more cells towards inner side, and few cells towards outer side. Few cells towards outer side, more cells towards inner side, few cells towards outer side. The cells which are produced towards inner side transformed into secondary xylem, also called as wood. The cells which are produced towards inner side, more cells are produced towards inner side, transformed into secondary xylem, that is also called as wood. Whereas few cells produced towards outer side, the few cells which are produced towards outer side, the few cells which are produced towards outer side, transformed into secondary phloem, also called as bast. The few cells which are produced towards outer side, transformed into secondary phloem, which is also called as bast. Like this, the cells of vascular cambium divides periclinally, mitotically, and produced more cells towards inner side. They transformed into secondary xylem, also called as wood. Few cells produced towards outer side, they transformed into secondary phloem, also called as bast. Vascular cambium not only produced secondary xylem and secondary phloem, vascular cambium also produce secondary medullary rays. Vascular cambium also produce secondary medullary rays. The vascular cambium produced not only secondary xylem, not only secondary phloem, and also produced secondary medullary rays, which are meant for lateral conduction in secondary body. In secondary body, the radial conduction or lateral conduction promoted by these secondary medullary rays. Altogether, that ring which is formed by the fusion of intrafascicular cambium and interfascicular cambium, that ring is called as vascular cambium. That vascular cambium produced secondary xylem, that is wood, secondary phloem, that is bast, and secondary medullary rays. These secondary medullary rays are meant for lateral conduction in secondary body. In cold and temperate regions, the activity of vascular cambium influenced by seasonal fluctuations. The activity of vascular cambium influenced by seasonal fluctuations in cold and temperate regions. For example, in spring season is the season of reproduction. There is a heavy demand for water. That's why in spring season, more number of xylem vessels are formed with wide lumen. More number of xylem vessels with wide lumen. This wood formed in the spring season is called as spring wood, which is also called as early wood. Spring wood also called as early wood. Whereas in autumn season, there is no much demand for water. There is no much demand for water because it is the leaf fall season. That's why in autumn season, less number of xylem vessels are formed with narrow lumen. Less number of xylem vessels are formed with narrow lumen. This wood means secondary xylem. This wood formed in autumn season is called as autumn wood, which is also called as late wood. So, in a single year, two types of woods are formed: spring wood, also called as early wood; autumn wood, also called as late wood. These two woods formed in a single year are present in two concentric rings. It is the spring wood and it is the autumn wood. Spring wood is called as early wood, autumn wood is called as late wood. These two woods formed in a single year present in two concentric rings. These two concentric rings formed in 1 year together called as annual ring, also called as growth ring. Annual ring also called as growth ring. Annual ring means it is not single ring. Two woods formed in a single year present in two concentric rings, spring wood followed by autumn wood, both are present in concentric rings together called as annual ring, also called as growth ring. One annual ring is equal to 1 year of the tree. One annual ring is equivalent to 1 year of the tree. 1 year of the tree. Like this, we can estimate the age of the tree by counting the annual rings. We can estimate, but it is not the correct method. It is not the correct method, but we can estimate the age of the tree by counting the number of annual rings. Number of annual rings. Annual ring means spring wood of the 1 year, autumn wood of the same year, present in two concentric rings, together called as annual ring, also called as growth ring. One annual ring is equivalent to 1 year of the tree. Like this, estimating the age of the tree, by counting the number of annual rings is called as dendrochronology. It is called as dendrochronology. Dendrochronology is the branch of botany. Dendrochronology is the branch of botany estimating the age of the tree by counting the number of annual rings. Like this, every year, first spring wood followed by autumn wood. The two woods, wood means secondary xylem, formed in the same year, present in two concentric rings, together called as annual ring, also called as growth ring. One annual ring is equivalent to 1 year of the tree. This branch of botany that deals with estimating the age of the tree by counting the number of annual rings is called as dendrochronology. Suppose it is the vascular cambium. This ring is vascular cambium ring. As I mentioned already, vascular cambium dividing periclinally, mitotically, producing more cells towards inner side. They transformed into secondary xylem, produced few cells towards outer side. They transformed into secondary phloem. The wood which is nearest to vascular cambium, this wood is called as sapwood. The wood which is nearest to vascular cambium, the wood which is newly formed from vascular cambium, the wood which is peripheral is called as sapwood. It is the peripheral wood, just inner to vascular cambium. It is newly formed wood. That's why just it is inner side to vascular cambium. This wood is called as sapwood. Sapwood is having vessels with wide lumen vessels with wide lumen. Sapwood is promoting conduction of water and minerals in secondary body. In secondary body, conduction of water and minerals by sapwood only. Sapwood is the newly formed wood, just inner to vascular cambium. Vessels are having wide lumen, and this sapwood promoting conduction of water and minerals in the secondary body. This sapwood also called as alburnum. Another name of sapwood is alburnum. Alburnum, that is sapwood present inner to vascular cambium. When the activity of vascular cambium is in progress, the sapwood which is nearest to vascular cambium pushed to the center. This sapwood pushed to the center. When the sapwood pushed to the center, now this wood is called as heartwood. This wood is called as heartwood. Heartwood is present in the center of the stem. Sapwood gradually transforming into heartwood. Vascular cambium producing sapwood, that is nearest to vascular cambium, that is just inner to vascular cambium. When new sapwood is coming, the already present sapwood transformed into heartwood and pushed to the center. That's why the wood that is present in the center of the stem is a heartwood. This heartwood vessels are filled with various types of substances like resins, gums, latex, etc. These vessels are filled with resins, gums, mucilage, etc. So these vessels are not useful for conduction. Moreover, these vessels are filled with tyloses. Vessels of heartwood are filled with resins, gums, mucilage, and vessels are having tyloses. That's why there is no chance for the conduction of water. Moreover, these vessels are more lignified. So heartwood providing mechanical support. It is not meant for conduction of water and minerals. It provides more and more mechanical support. Moreover, it is highly durable. It is providing mechanical support. It provides more durability. That's why heartwood also called as duramen. Sapwood vessels are having a wide lumen and they are meant for conduction, and in secondary body sapwood only provides conduction of water and minerals. Sapwood also called as alburnum. Sapwood gradually converting into heartwood pushed to the center of the stem, and this heartwood having vessels filled with resins, gums, mucilage, tyloses, and more lignified. This wood is not meant for conduction. It provides mechanical support. Like this, the secondary growth is going on inside the steel due to the activity of vascular cambium. When this intrastellar secondary growth is going on, steel increases in diameter. When the steel is increasing in diameter, pressure developed on the cortex and epidermis, which are outside to the steel. Outside the steel, cortex is present in the primary body. Outside the cortex, epidermis is present in the primary body. Pressure developed on cortex and epidermis, they ruptures. They ruptures. So, there is a need of extrastellar secondary growth, means secondary growth outside the steel, that is called as extrastellar secondary growth. Extrastellar secondary growth. Secondary growth outside the steel is called as extrastellar secondary growth. In case of extrastellar secondary growth, cambium formation occurs in the cortex. The cambium that is formed in the cortex region is called as cork cambium. This cambium is called as cork cambium. How? The vascular cambium promoted the secondary growth inside the steel. Cork cambium promotes secondary growth outside the steel. Cork cambium formation occurs in the region of cortex. In the region of cortex, cork cambium formation takes place. Cork cambium is lateral in position. Cork cambium is lateral in position, and cork cambium is secondary in origin, and the function of cork cambium is secondary growth. Cork cambium that is lateral in position, cork cambium that promotes secondary growth outside the steel, it is secondary in origin. Why I called it as secondary in origin? All of you know the cortex parenchyma present in the cortex undergoes dedifferentiation and produced cork cambium. That's why cork cambium is secondary in origin, lateral in position, and the growth is extrastellar secondary growth. As usual, cork cambium divides periclinally, mitotically. Cork cambium divides periclinally, mitotically, and produce more cells towards inner side, few cells towards outer side. The more cells produced towards inner side from cork cambium transformed into secondary cortex. The few cells produced towards outer side. Transformed into cork. Cork cambium produced more cells towards inner side. They transformed into secondary cortex and produced few cells towards outer side. They transformed into cork. Cork cambium, secondary cortex. Cork also called as phellum. Cork cambium also called as phyllogen. Secondary cortex also called as phylloderm. Cork is called as phellum, cork cambium called as phyllogen, secondary cortex called as phylloderm. All together, these three together called as periderm. Periderm is formed by cork, cork cambium, secondary cortex. Cork also called as phellum, cork cambium also called as phyllogen, secondary cortex also called as phylloderm. Altogether it is called as periderm. Altogether it is called as periderm. Let us take cork first. Let us take cork first. Cork is made up of dead cells. Cork is made up of dead cells. Walls are deposited with suberin. Walls are deposited with suberin. Cork is made up of dead cells and walls are deposited with suberin. Cork provides protection to the secondary body. Cork provides protection to the secondary body. Cork is made up of dead cells. Dead cells. Walls are deposited with suberin. But at some places in cork, at some places, living cells are also present. In cork region, at some places, living parenchyma cells are also present. These living parenchyma cells are called as complementary cells. These living parenchyma cells are called as complementary cells. The region where complementary cells are present, that region is called as lenticel. That region is called as lenticel. Lenticel is the region. It is present in lens shape. That's why it is called as lenticel. Lenticel is not cell. Lenticel is an area, lens shaped area. This lenticel having many powdery living parenchymatous cells. These powdery living parenchymatous cells are called as complementary cells. These cells are called as complementary cells. In the primary body, all of you know, stomata are present for exchange of gases. Stomata also promotes transpiration, but now it is in secondary body. There is no epidermis, so no stomata. But what about exchange of gases? Yes, lenticel, which is made up of complementary cells meant for exchange of gases in the secondary body. In secondary body, lenticel promotes exchange of gases and also little bit transpiration, just little bit transpiration or negligible transpiration. That is about cork. It is a cork cambium, also called as phyllogen. All of you know very well. It is the cambium meristematic tissue dividing periclinally, mitotically and producing secondary cortex towards inner side and cork towards outer side. So cork cambium producing cork towards outer side and secondary cortex towards inner side. Secondary cortex is made up of parenchyma cells having chloroplast and the function is assimilation, carbon assimilation, nothing but photosynthesis. So secondary cortex, that is just inner side to cork cambium, having parenchyma cells filled with chloroplast and meant for carbon assimilation. So after completion of secondary growth in the dicot stem, let us imagine 2 years old dicot tree. 2 years old dicot tree. In the center of 2 years old dicot tree, spring wood of first year is present. Outer to spring wood of first year, autumn wood of first year is present. Spring wood of first year, autumn wood of first year together form first annual ring. First annual ring. Autumn wood of first year surrounded by spring wood of first year. Spring wood of first year surrounded by a concentric ring of autumn wood of second year. Second year. Autumn wood of first year surrounded by spring wood of second year surrounded by autumn wood of second year. Spring wood of second year, autumn wood of second year together constitute second annual ring. I already mentioned it is the 2 years old tree. First year, second year, autumn wood of second year surrounded by vascular cambium. Vascular cambium. And all of you know vascular cambium produce secondary xylem, also called as wood, towards inner side. Secondary phloem, also called as bast, towards outer side. Secondary phloem, also called as bast. Secondary phloem concentrically surrounded by secondary cortex, outside to secondary cortex or peripheral to secondary cortex. Cork cambium is present. Outside the cork cambium, cork is present with lenticels. Cork is present with lenticels. It is the secondary body. It is the secondary body from peripheral to center, margin to center, centripetally. Cork, cork cambium, secondary cortex, secondary phloem, vascular cambium, autumn wood of first year, spring wood of second year, autumn wood of second year, spring wood of second year, autumn wood of first year, spring wood of first year. I already explained the wood that is nearest to vascular cambium, that is inner to vascular cambium, just inner to vascular cambium is called as sapwood, that is called as sapwood. Sapwood promotes conduction in secondary body, I already mentioned. So in 2 years old dicot tree, in 2 years old dicot tree, autumn wood of second year is nothing but sapwood. Autumn wood of second year is nothing but sapwood. The remaining 3 woods, spring wood of first year, autumn wood of first year, spring wood of second year, these 3 together heartwood. Together heartwood. The wood nearest to vascular cambium, sapwood, that is autumn wood of second year, in 2 years old dicot tree. The remaining 3 comes under heartwood, spring wood of first year, autumn wood of first year, spring wood of second year. Heartwood, as I already explained, it provides mechanical support, it doesn't useful for conduction. It is the secondary body. If you observe, entire tissue present above the vascular cambium, entire tissue present above the vascular cambium, don't include vascular cambium, entire tissue present above the vascular cambium together called as bark, together called as bark. Entire tissue present above the vascular cambium together called as bark, bark. So bark includes, come on, cork plus cork cambium plus secondary cortex plus secondary phloem. Secondary phloem. Bark includes cork, cork cambium, secondary cortex, secondary phloem. The entire tissue present above the vascular cambium is called as bark. Bark includes secondary phloem, secondary cortex, cork cambium, cork. As I already explained, cork, cork cambium and secondary cortex together called as periderm. These three together called as periderm. So, bark, in another words, periderm plus secondary phloem. Periderm plus secondary phloem. Periderm plus secondary phloem together called as bark. As I mentioned already, cork, secondary cortex produced from cork cambium. Secondary phloem produced from vascular cambium. So altogether, bark is the combined product of vascular cambium and cork cambium. Bark is the combined product. Bark is the combined product of vascular cambium and cork cambium. Suppose if you take a dicot tree, remove bark at one place, remove the bark at one place in the form of a ring, remove the bark at one place in the form of a ring, that experiment is called as girdling experiment or ringing experiment. Remove the bark at one place in the form of a ring, that experiment is called as girdling experiment, also called as a ringing experiment. Then you imagine what happens. There is no problem for the conduction of water and minerals, because sapwood meant for the conduction of water and minerals present inner to vascular cambium. What you removed is the bark, that is the tissue present outside the vascular cambium. So there is no problem for the conduction of water and minerals. Leaf synthesized the food, leaf prepared, photosynthesis, they came down at the region of a ring where you removed the bark in the form of a ring. Secondary phloem is also gone. Secondary phloem is a pot of bark. As I mentioned, secondary phloem is a pot of bark. So in that region, secondary phloem is also gone. Food doesn't move downwards. It accumulates above that ring. So the region above that ring swells. The region below the ring doesn't receive the food material. It dies gradually. After some years, total plant also dies. First, the pot below the ring dies. Later, total plant dies. This experiment proves that the phloem is responsible for the conduction of water and minerals. Phloem is responsible for the conduction of water and minerals. So, in primary body, as you already maybe studied, in primary body, epidermis, hypodermis, cortex, the innermost layer of cortex is endodermis, stele, in stele pericycle, medulla, vascular bundles, all you already observed. But it is the secondary body where you cannot see all these regions. In secondary body, the peripheral region is cork, inner to cork, cork cambium, secondary cortex, secondary phloem, all these together called as bark, vascular cambium, inner to vascular cambium, secondary xylem, also called as wood, that is nearest to vascular cambium, sapwood, in the center of the stem, heartwood. These are the different regions present in secondary body of dicot stem. Dicot root also shows secondary growth. Dicot root also shows secondary growth, but very small difference between secondary growth of dicot stem and that of root. In case of dicot stem, you observed how the vascular cambium formed. Already intrafascicular cambium inside the vascular bundle, that is primary in origin. In between vascular bundles, primary medullary rays are present. They undergone dedifferentiation and produced interfascicular cambium. Both intra and interfascicular cambiums are combined and formed vascular cambium. Vascular cambium is partly primary, partly secondary in origin in case of dicot stem. In dicot stem, vascular cambium is partly primary, partly secondary, whereas in the secondary growth of dicot root, total vascular cambium newly formed, is newly formed from the tissue present in pericycle and also from the tissue present just below the phloem. The tissue of pericycle, the tissue present just below the pericycle produced vascular cambium. So in dicot root, vascular cambium is completely secondary in origin. In dicot root, vascular cambium is completely secondary in origin. Moreover, initially it is wavy. Because pericycle involved, tissue present below the phloem involved in the formation of vascular cambium. So vascular cambium in dicot root initially wavy, later it becomes round and starts secondary growth. So the basic difference between secondary growth of dicot stem and dicot root, in dicot stem vascular cambium is partly primary, partly secondary in origin, whereas in dicot root vascular cambium is completely secondary in origin. In dicot stem, vascular cambium from the beginning onwards it is round, whereas in dicot root vascular cambium initially wavy and later it becomes round. These are the basic differences between secondary growth of dicot stem and that of dicot root.